Yeah, okay. Thanks very much. Good morning, everyone, and thanks for the invitation to give a talk here. So why should we look at brain networks at all? There are different reasons. One reason, if you look at artificial intelligence, is that AI systems are still not as intelligent as humans, even though they can be very intelligent in very specific topics, but they can't really generalize and reason as humans can do. So there's still limits in terms of what AI systems can do at the moment. And AI systems still today are organized in a different way compared to the human brain. Okay, so there's still many features of the human brain that are not implemented in neural networks. And one person who already thought about a comparison between computers and brains was John von Neumann, who was the architect of the modern computer architecture with working memory, a central processing unit, and so on. And in his own words, because of low speed and accuracy of calculations, large and efficient natural automata, so those are brains, are likely to be highly parallel and have low logical depth, a low number of processing steps. The reasoning is that biological systems are always noisy, so they always introduce a certain error. So if you have 20 processing steps and each step is introducing 5% error, then the end result will be totally wrong. So you have to have as few processing steps as possible. And I was giving an update in 2007 how we can actually see this if we look at brain connectivity. Another reason to look at brain connectivity is that neural networks, biological neural networks, can be very robust. So one example here is from animal experiments. You can see a slice of the spinal cord, and 95% of the spinal cord was cut. So it's only the small shaded area, 5%, that is still remaining. And still, the rodent was, after a few weeks, recovering and walking again. So not walking as before, limping a bit, but able to walk, even though 95% of the fibers are gone. Another example here is an 11-year-old boy who had severe epileptic seizures. And because of that, almost all of one hemisphere was removed. So you can see the white parts here, which is just cerebral spinal fluid. And a bit of visual cortex is still there. So even though half of the brain is gone, the patient fully recovered, got a driving license, high school degree, and so on, even though half of the brain is missing. You can't do that to computer networks easily. So you can't just knock out half of the network and have this kind of recovery. So how are brains organized? First of all, we can look at different types of brain connectivity. If two nodes A and B are connected, there could be a structural connection. So this means that there is a physical connection between two parts of the brain. It could be a fiber tract between brain regions, or it could be an axon between individual neurons. We can also look at functional connectivity. And this means that there's a similar activity pattern in different parts of the brain. So you can look at the time series, if you stick electrodes into the brain, or if you look at fMRI. And if there's a high similarity between the time series, a positive correlation, you would say that there's a functional link between those parts of the brain. And finally, you can look at effective connectivity. And this means that there is a causal relationship between two parts of the brain. So there could be a link going from A to B, and this means that A is modulating activity in B. So if activity in A is increasing, then activity in B is increasing. Or if activity in A is increasing, then activity in B is reducing. So it can be synergistic or antagonistic. So you can also look at effective connectivity. Looking at effective connectivity is a bit more complicated. So you have Carl Frissen's work with DCMs, but normally you only look at relatively small networks. So you might only have 10 or 20 nodes in your network, and then you can look at those causal relationships. But if you want to look at large-scale networks, if you have 100 nodes or 1,000 nodes or 10,000 nodes, you would basically look at just the structure or functional connections. So this is what you see. Is there any way to get this out of the way? One moment. So this is what you see when you open up a new science textbook. This is the visual system. So you can see that information starts in the retina, then goes over the lateral genicular nucleus, and then enters the cortex V1, goes into a ventral pathway, which is for the position of objects. No, is for the recognition of objects, and the dorsal pathway for position and movement. But this is only a simplified version of the visual system. This is the real visual system. So the layout here is like a layout for a microchip. So you can see that there are a lot more connections between different parts of the visual system than you would normally see in a textbook. Can any one of you immediately see the difference between dorsal and ventral pathway? No, me neither. Okay, but you can do statistics and you can still segregate those pathways looking at this map, but it's not as obvious as in the new science textbooks. And this is only the visual system. So we can look at connectivity in the whole brain, and it gets even more complicated. You can parcellate the brain into 100 regions or 300 regions, depending on your parcellation scheme. What you see here is an image generated from diffusion tensor imaging. That's a special MRI protocol where you can reconstruct fiber tracts in the brain. The color gives you the local direction of the fiber tract. So green is from the back to the front, or from the front to the back. Blue is from the top to the bottom, or the bottom to the top. And red is left, right, or right, left. So you can see the corpus callosum up there. You can see the fibers going up and down the spinal cord. One problem with this data is that it doesn't tell you about the directionality of those fibers. Okay. So from the data, they all look bidirectional, but they're not all bidirectional. We know from studies in monkeys where you are not using MRI, where you basically dissect the brain, that there were some unidirectional links in the brain. And we know from other animal studies that there are unidirectional links. So there might be some one-way streets in the network, but we can't see them with the methods that we have at the moment. It's something to keep in mind. We can look at this network, and we can already predict the age of the subject, ±8 years, and the IQ, ±3 IQ points. So just looking at the physical structure of the network without any traffic, without any information spreading, you just look at what highways are there in the brain, how wide are the highways, which means how thick are the fiber tracts. Just based on this information, you can predict IQ and age. But the idea is that we can use this as a starting point to predict how to treat patients in the hospital. So if we have mental health disorders, or if we have dementia or Parkinson's disease, what would be the best approach to treat patients? If we look at the architecture of networks, we can distinguish different archetypes. You have Erdős-Renyi networks, which are random networks. So you just randomly connect regions. So in this case, regions are all arranged on a circle, and you just randomly add links between regions. You could have a scale-free network where you have some highly connected nodes in the network. So for example, deep brain structures like hippocampus, amygdala, those are all highly connected structures of the brain. You could have a regular network where you have just connectivity between neighbors, a small world network where you also have some long-distance shortcuts that help to reduce the number of steps you have to go from one node to another. You could have a modular network where there are many connections within a module and few connections to other parts of the brain. Or you could have a hierarchical network where you have modules, and then you have modules within modules, and then modules within those modules, and so on. So you have a nested architecture of modules. And one thing to keep in mind is that the brain has all of those features. So it's not just, you can't just say, okay, the brain is a small world network, but then it also has hubs, and it also has modules, and so on. So the brain has a mixture of those features, a combination of these archetypes. And the structure of the brain follows the function of the brain. So we can look at those different archetypes again. There could be a regular network, a modular network, a hierarchical network. And we find this at different stages of evolution. If we go to simple organisms like Cnidaria, they have two stages. One stage is they're just attached to the seafloor, and then they're streaming food with those tentacles into the stomach. And the other stages, they are floating around in the water. But during this stage here, you basically have a nerve net. So the neurons are on the outside of the stomach, and they're just connecting neighbors next to each other. So for this stage, for this function, you just have a nerve net, a regular network. If we then go to different organisms like C. elegans, a small roundworm around 1 to 1.4 millimeters long, you basically get modules because you have a functional specialization. So over here, there's only one function contracting the stomach, so getting water in and out. It's like the heart muscle just contracting. But if we go to C. elegans, you have different clusters of neurons called ganglia. Those are the modules, and they have different functions in the animal. If we then go further during evolution, we get to the human brain or the monkey brain, and there you have hierarchical networks. So you have one cluster or module in the network responsible for vision, but then you have modules within the visual system. As I mentioned earlier, one module for object recognition and one for movement of objects. So the more complex the task of the brain, the more complex is the network. So we can look at different species and we find similar architectures in those species. We can look at the fruit fly, the mouse, or the rhesus monkey, and we find a similar network architecture. Some of the features that we find is first, this imbalance in connectivity. And again, this is something we don't see if we have MRI diffusion sensor imaging. So most of the time, you have a strong connection in one direction and a weak or even absent connection in the other direction. And this imbalance gives more degrees of freedom. So there are more degrees of freedom in order to tune your network if you can adjust one of the two weights independently. Another feature is the modular organization. So we have functional specialization and we have modules responsible for different brain functions with many connections within a module and few connections between modules. And we also have an architecture with some highly connected nodes, like amygdala, hippocampus, thalamus. So you have highly connected nodes in the network. And you also have stronger connectivity between those highly connected nodes or hubs. And this is called the Witch Club network. So there are more connections between hubs than you would expect by random wiring. And because of the Witch Club network, you can easily reach the hub in one module. Then information goes to another hub in another module, and then can broadcast to the other module. So it's a shortcut, a super pathway for basically spreading information in the system. If we look at brain disorders, they often affect those highly connected nodes. So if you have a node which is not highly connected, there might not be a big impact on cognitive function. But if you have a node that is highly connected, like hippocampus for Alzheimer's disease, you have a big impact on the spreading of information in the system. So we have this hierarchical connectome organization. And hierarchy can mean different things to different people. Okay, so you might say, okay, hierarchy means that you have different levels of processing. So you have a sequential hierarchy where you have the starting point, early visual processing, and then you have a higher visual processing later on. A hierarchy can be topological. And that's what I was talking about earlier, where you have modules, and then you have modules within modules, and modules within those modules, and so on. It can be temporal. So you can use the time axis as a measure of your hierarchies. You can zoom in and out with your temporal resolution. So if you have a relatively high temporal resolution, you might see some frequency peaks in brain activity. But if you lower the temporal resolution, it just all averages out. Okay, so you could have a hierarchy in terms of the time scale. And you can have a hierarchy in terms of the spatial scale. So you can look at the whole brain, or you can spatially zoom in into a region, into a column, into a layer, and so on. So to go beyond the standard approach of just looking at the brain at a low resolution, we're basically zooming in topologically. We're not only looking at a network between brain regions, but also at a network within brain regions. In order to do that, we parcellated the brain into 50,000 nodes. So you can see that we have the cortical surface, and we are just forming 50,000 triangles on the cortical surface. And we then look at connectivity between those triangles. So because of that, we also have connectivity within the standard brain regions. We might have 100 to 1,000 nodes within each brain region.
Here, connectivity, you mean
Structural connectivity. Yes, yes. So physical connectivity. Okay, now comes the tricky bit. So let's see what. So we can basically fly through this connectome. Here you see the whole brain. We can zoom in into one hemisphere, and we can zoom in further and further. And what we then see at the local level, just in the middle of the screen, is one brain region where you can clearly see there are two different modules within that brain region. So we have a modular architecture within brain regions as well. It's kind of a self-similarity of the network. This is showing the same thing again. We have the self-similarity and some scaling laws here. So the idea is that the self-similarity might indicate what is going on within brain regions. Okay. So for example, if we think about the motor region, there is one part of the motor region responsible for arm movements and another part responsible for leg movements. And the idea is that those are different modules if we look at structural connectivity. So one module for arm movement, one module for leg movement. So we're currently testing that with fMRI data to see whether we really are activating those different modules for different tasks. But we first tested whether the connectivity is an artifact of the reconstruction or not. In order to do this, we can look at two brain regions here. With, you can see the internal modular architecture, left superior frontal and left superior parietal area. And we then look at connectivity between those two regions. And if there would be an artifact, then there would just be some random connectivity between those regions. But what we basically see is that a module in one region is connected to a module in the other region. Okay. So it's a non-random architecture. So it's like you have one module for sensing on the arm, and that's connected to a module for moving the arm. Okay, so you have this specific architecture that modules in one region are connected to modules in another region. And those modules we find are also spatially organized. They're just adjacent patches of tissue. So you see an example here, left superior frontal cortex, where the modules within a brain region that we find are just adjacent to each other. So that makes sense if you think that patches of cortical tissue have the same function. And this is what we see for the left postcentral gyrus. And below, this is from another paper, Yeo and colleagues, and they looked at activation of the motor system. And you can see those patches of tissue that are activated for tongue movement, hand movement, and foot movement. So potentially, those patches correspond to the modules that we see with our structural network reconstruction. But we're currently testing that in more detail. So what? Yes.
You talk about power module, etc. But what about inside those? Is it completely homogeneous and no modules? So do you see also modules inside the foot movement area?
We don't have the resolution for that because so for the, let's say, food movement area, we might only have 20 nodes or so. So it's not enough to look at the modular architecture of that small network. Possibly if we would have better data, so this is all based on the 3 Tesla MRI system. We're currently getting an 11.7 Tesla MRI system in Nottingham. So with that spatial resolution, we might be able to learn more about this local organization, but not with the data we have at the moment. Yeah. Okay. Yeah. So what information can this actually give us? In order to see whether there's any clinical impact of high resolution, we looked at temporal lobe epilepsy. For temporal lobe epilepsy, in 1/3 of the cases, pharmaceutical drugs are not working, and then patients often undergo brain surgery. But unfortunately, in 1/3 of the surgery cases, the surgery doesn't work and people still have epileptic seizures. So we wondered, can we actually predict whether someone will have epileptic seizures after surgery or not? So we looked at 33 patients, 36 controls, and we looked at the 2 different types of networks. One is to look at networks within brain regions, and the other idea is to look at networks between brain regions, which is the standard way to look at brain connectivity. So we either have at the top a network within one brain region. So you can see an example within the fusiform gyrus, or we look at the network between brain regions, the standard approach. And we basically find a better performance in terms of predicting surgery outcome by using this high resolution network, by looking at connectivity within brain regions. You can see the area under the curve here. But the important point is this: the specificity is 90% compared to only 75% with the standard network. So local changes are better in correctly predicting bad outcomes. Yes?
It depends on the brain region. Because like you can operate in different brain regions, now you know how it correlates or not. It's accuracy if you do it in different brain regions.
No, we did it in all brain regions.
Yeah.
Yes, yes. So we didn't single out a particular brain region. We looked at the whole brain, but only the within region network.
But you didn't see like within regions or regions where you have like better predictions than others.
No, we didn't do that. No. Yeah. So we can basically predict bad outcomes. Bad outcome means the patient has surgery, but there was no positive effect of surgery. So the patient still has epileptic seizures after surgery. So it would be useful to predict that before surgery because of all the risks involved in opening the skull. So future questions would be, are local network features better predictors for what region should be targeted in surgery or for invasive and non-invasive brain stimulation, other diseases? And also, are those local networks better biomarkers of diseases? So for example, if you think about schizophrenia, normally occurs between, let's say, 18 to 30. Can we actually predict that a few years before onset? And local networks might show an earlier change compared to the global networks. And same for Alzheimer's disease. So it might be something to look into, to look at those high resolution networks as a predictor of later disease onset. I talked a lot about topology, but it's also important to look at network dynamics. So brains are nonlinear systems. Small changes can have large effects on system behavior. If we look at those different connectomes here, you can see the bottom 2 look quite similar topologically. But if you look at the behavior of those brains, you would have seizure in the first and in the third one. So you would classify them differently. And the reason is that small changes in the topology can have a big impact on the dynamics of the network. If you use machine learning, and machine learning is just based on the structure of the network, you would just cluster the bottom 2 into the same cluster with machine learning. But the idea is that because you have small changes in the topology, they might lead to big changes in the dynamics. So it's not enough to only look at the topology of the network. So there's a need for simulating brain dynamics. So this is one example where we simulate brain dynamics for epilepsy. We use a dynamical model for each brain region called the Benjamin model. And this model is basically oscillating, and you can see that over time, the oscillations get larger and larger until they reach the full amplitude. We call that the escape time. So that's basically the time when that brain region shows epileptic activity. So we can simulate activity in the whole brain. The coupling between regions is based on the physical fiber tract network. And we can then see that some brain regions start to show this epileptic activity earlier than others. So we would predict that those must be starting points of epileptic seizures in that particular patient. Those must be regions that we can remove in order to avoid seizures in that patient. So this is a different paper published in Brain in 2017, where we took this approach of modeling dynamics in the system. In this case, we have implanted electrodes. So you can see a grid of electrodes just sitting on top of the cortical surface. And in this case, we can look at functional connectivity between the electrodes. If you have a similar time series between two electrodes, you have a functional link between those electrodes. And we then have a computational model that is simulating activity in the system, and again, looking at the escape times in the computer model. But the interesting bit of this model is that you don't need any seizure activity to establish the model. So what you do with patients in the hospital is you implant the grid, and then the patients are in the hospital for a couple of days, maybe a week or so, until you have a sufficient number of seizures to predict where to do surgery. But for our model, we don't need seizures at all. We can just have the patient in the hospital for half a day, and that's all the data we need, or a couple of hours. So we don't need a full week in order to run our model. We don't need any seizure events for this. So we can then predict whether surgery is effective or not. On the left, you see one patient with a seizure-free outcome. And you can see that the location of surgical resections under the black electrodes is exactly the same region that we would predict in our computer model to be the starting point of epileptic seizures. So you have a complete overlap, and that patient is seizure-free. For the patient on the right, you have no overlap at all between the part of the brain that was resected under the black electrodes and the part in red where we would predict that's the starting point of epileptic seizures. And that patient still had seizures after surgery. If we look at all the patients in the cohort, if our computer model predicts that the patient is seizure-free, then in 100% of the cases, the patient is seizure-free. If the computer model predicts that there will still be seizures afterwards, in 27% of the cases, the patient is seizure-free, and in 73% of the cases, the patient is not seizure-free. So this kind of model can, on the one hand, give a prediction whether surgery works or not, but it can also indicate what other region might need to be removed. So if you look at the right-hand side, the region in red, this would be a region to consider for that particular patient to remove. And then surgeons can look into that region in more detail and consider whether they want to remove that one rather than the standard one they would normally go for. So dynamics are very important, but dynamics are driven by the spatial organization of the brain. And I will make the point that the spatial organization is underrepresented in the brain network community in terms of looking at spatial features, but it's very important for brain function. We can look at the spatial organization in different species, let's say the rhesus monkey. I don't know if anyone of you has seen rhesus monkeys before. They're like the size of cats. Okay, so not. And we can look at C. elegans. So there are around 304 neurons in C. elegans, but we have the network for 277 here. You have on the left the head of the animal, then the kind of spinal cord. I mean, there's no spine, but basically you have those fibers going down the belly of the animal. Later during evolution, it swaps around. So we have the cord at the back, but for the elegance, it's in the front. And then the tail of the animal. And we can zoom in, we can basically look at the ganglia in the head. And we can then look at how long are connections in the system. So you can measure the length of fiber tracts in the rhesus monkey. See the distribution here. So you have many fiber tracts that just go between nearby brain regions, but then you have some fiber tracts that go over a long distance, let's say from the back of the brain to the front of the brain. You see the same when you zoom in. So you can zoom in into one brain region, into one column, and into one layer. So in this case, you have two layers, layers 2, 3 in the red. And again, you have a preference for short distance connections to nearby neurons, but then you have some connections that go further. And of course, you have connections that are not shown in the cystogram that go out of the area to other brain regions. But basically, normally 60 to 80% of the connections of a brain regions are internal. They don't go to other parts of the brain. So you have lots of internal communication. You see it again if we look at C. elegans. If we look at the head of C. elegans, the preference for short distance connections. If you look at the whole of C. elegans, you also have long distance connections that almost go to the full length of the animal. So it would be 1.4 millimeters long. You can see some connections that go almost all the way. Of course, we would get a similar picture in humans if we include the spinal cord, because you have long, 1 meter long connections going through the spinal cord, longer connections going through it, but they're not part of the normal analysis. So we see similar features across species. One question could be, why do we see this exponential tail? So before we can basically model this distribution as a gamma distribution, but in all cases, you have this exponential tail. So the further away, the lower the probability to establish a connection. And we can basically explain this through a random process. So let's say X is a number of steps until another neuron is in the range of an axonal growth cone. So you have a growth cone and an axon basically migrating through the tissue. P is the probability that the space that is entered by the axon is having a neuron inside, and Q is probability that the space is empty. So let's say in the first step here, the axon is growing into a new space, it's empty. A second step, still empty space. Third step, there's a neuron, so a connection is formed. So basically, probability to form a connection after n steps or after spatial distance n is 1 time the probability to hit a neuron, that's the last step, and n minus 1 times the probability that the space beforehand is empty. So this can basically explain how you get this exponential decay over distance. So it is a bit like walking through a room with people, and it's much more likely to bump into someone early on than going through the whole room without bumping into anyone. Okay, so you get this higher probability for short distance connectivity. So we can see that most connections are over a short distance. And this has led to the idea that the brain should minimize the amount of wiring. So minimizing the total wiring length reduces the metabolic cost for establishing connections and also for propagating signals over a long distance. So this has led to the idea of component placement optimization, CPO, proposed by Christopher Cherniak in '94. So he's a philosopher based in the engineering department, which might be the reason why he was looking into computer chip design and arguing that the brain might be organized in a similar way. For computer chips, you're basically minimizing the amount of wire you have on the chip, because the more wire you have, the more heat dissipation and the more energy loss you have. So you want to have wiring on a computer chip as small as possible. So Cherniak was arguing that the brain is exactly the same. Component placement optimization means that there is no way to get a better spatial layout of the brain. Whenever you swap around two areas, the total length of the connection is getting longer than it was before. So the original organization is always the best one. As an example here, you have this connectivity on the left side. You swap around nodes A and D. So they retain their connectivity. You just swap around their spatial position. And you can see that the total length of wires is longer on the right-hand side. So the idea was that this happens all the time. There's no way to get any better outcome. But that's not the case. So I was looking at new data sets. So at that time, Cherniak didn't have information about the spatial position of nodes. And looking at this new data set, I found that it is possible to get better wiring layout. For the rhesus monkey, we can reduce the total length of wires by 30%. For C. elegans, we can reduce it by 50%. So what do we see if we look at the connectivity before and after optimization? What you see at the top is the arrangement after reducing total wiring length. So when we basically rearrange the position, and if you look at the distribution here compared to the original one, you can see that those long distance connections, they just disappear after rearranging the network. So before, we have a lot of long distance connections that are non-optimal. So it is a bit like having an optimal network and then randomly sprinkling long distance connections on top of it, which makes it non-optimal. So for some reason, the brain has more long distance connections that would be beneficial for energy saving. But those long distance connections are beneficial in other ways. So we can look at the characteristic path length or average shortest path. And this is the average number of connections you have to cross to go from one node to another. Okay, so how many fiber tracks do you have to cross to go from one brain region to another? And we can then generate networks that don't have long distance connections at all. So in this model, I'm not rearranging the position of regions, but I'm rearranging fiber tracks in the network in order to reduce the number of long distance fiber tracks. So that's a minimal configuration compared to the original. And when we get rid of those long distance connections, we see that the path length is increasing for the rhesus monkey and for C. elegans. And the reason is that long distance connections are providing shortcuts in the system. So if we want to go from the back of the brain to the front of the brain, we might go from this node to this node, LIP, and then frontal eye field, and then we are here. So we can go there quickly. So if we don't have long distance connections, we have to go from this region to this region, from this region to this one, from this one to this one, and so on. So we have a much longer path length, many more intermediate nodes. So long distance connections are shortcuts that help to reduce the characteristic path length. So there are fewer long distance connections, means there are longer path lengths. So what's the benefit of having few intermediate steps in your network? One benefit is that we can have synchronous processing in nearby and faraway regions because they receive information at about the same time. So there might be a few milliseconds delay because one fiber is longer than the other, but it's still more or less the same time for processing. We also have reduced transmission delays. If you look at, if you look at the best case scenarios, you have an incoming action potential and then the postsynaptic neuron is firing as a result of that, you still have a delay of 1 to 2 milliseconds that is introduced. So if you have more neurons in your pathway, you're increasing the delay for propagating the information. And you can avoid that if you have a direct long distance connection. You also have less cross-modal interference. So let's say you want to go from the back of the brain, visual cortex, to the front of the brain. If you only have short distance connections, you have to go through the sensory motor cortex, which will destroy your visual signal. But if you have a long distance connections, you can bypass those parts of the brain and go directly to other parts of the brain. You also have higher reliability of transmission. If you open up a new science textbook, it basically tells you that you have an action potential, and then you have vesicles, neurotransmitter release to the synaptic cleft, and then pick up by the postsynaptic neurons, and then postsynaptic neuron might fire. The sad reality is that in 50 to 90% of the cases, there's not even neurotransmitter release. So you have an incoming action potential, and there's no release of neurotransmitter. So often it needs a burst of spikes, a burst of action potentials to make sure that there is release of neurotransmitter into the synaptic cleft. So we have a high probability at each step that the signal just gets lost. And again, you can avoid that if you have a long distance connection. So we know that long distance connections are important, and we know from studies in humans that long distance connections are important. So they are linked to IQ, they're linked to processing for dementia patients, and so on. So if you get rid of long distance connections, you have cognitive deficits. So we were testing this idea for human structural connectivity as well. And what you see here is the change in total wiring length. We were using 280 subjects from the Human Connectome project, and again, optimizing the network layout. And we can see a reduction around 20 to 22% for the total wiring length. So that's far away from zero and far away from positive numbers. So for all 280 subjects, we are able to get a better spatial layout. So you may wonder, what's the benefit? We already mentioned the benefit in terms of providing shortcuts for the system. But then we also thought there might be a benefit in terms of network dynamics. We were simulating activity in those networks. So we took structural connectivity. Each brain region is represented by a Kuramoto oscillator. So we can set it to 10 hertz, 20 hertz, 40 hertz. So we just set it to a certain frequency. The coupling between oscillators is depending on the strength of the fiber tract. If it's a strong fiber tract, there's stronger coupling. And we also have delays depending on the length of the fiber tract. We can then measure the synchronization in the whole network. So you can see synchronization is fluctuating over time. We can look at the mean synchrony and the standard deviation metastability. So metastability is basically describing how strong the fluctuation is. Are we very close to the mean, or are we very far away from the mean in the network? If we compare the original network and the minimized network, where we swap around the position of brain regions, we find that for the minimized network, we have higher synchrony. Higher synchrony is not always good. We have higher synchrony during epileptic seizures. So it's not always a good outcome if you have high synchrony in the network. What we also see is that we have a much lower metastability, a much lower variability in those minimized networks. So if we swap that around, in the original brain, we have a higher variation in synchronization. And this higher variability might make it easier to switch between different states in the brain. Okay, so it's easier to get from one state to another if you have higher variation. And this might be another benefit, dynamic benefit of the network organization. Yeah.
It's like binary on the original. Do you have an explanation? Like, or it's just the outliers?
Oh, yeah. So yeah, could be. I think it's a bit misleading. I mean, basically, let's say 3/4 of the points are here, but it's a bit strange to see those two different areas. Yeah. Yeah. Yes. Yeah. Mm. Mm. Where basically shortening, where do you see the shortening of delay? Yes. Yeah. No. Shorter pathway means you have a lower amount of synapses. Shorter pathway means you have fewer connections to cross. Yes. Yeah. Yeah. Okay. Yeah. Okay. So we can basically test what the effect of different network features is. So in this case, we looked at, again, the Human Connectome project, where we also have IQ data. We have different subscores of the IQ you can see here. And we then tested whether those subscores of the IQ correlate with network features. We tested topological network features. We have the clustering coefficients, so how well are neighbors of a node connected, unweighted and weighted. The path length, again, unweighted and weighted, edge density. And you can see some correlation between IQ subscores and topological measures. But if we then look at spatial features of the network, you can see much stronger correlations between IQ and spatial features. So we have the convex hull volume, which is basically you wrap a foil around the brain and you just look at the total volume of the brain. So that correlates strongly. You have the distances between nodes. Okay, so let's say you have a node A connected to node B and C. You can measure how long is the distance between B and C. So are B and C nearby or are they further away? If they're nearby, A is integrating maybe similar information. If they're far away, then A might integrate different kinds of information, visual and auditory. And we're currently testing the Kuramoto model as well. And again, we find that the dynamic model is again better than the topological model as a spatial model you see here. So this indicates that the spatial organization is very important. And that's why I called it in the previous paper, the delayome. Okay, so not the connectome, but the delayome. The delayome is the set of delays in your network. And the delays in the network can influence how well the network is functioning. If you have demyelination, let's say for Alzheimer's disease, you have a lower speed across the fiber tracks, so the delays get longer, and then information can't arrive on time. Synchronization is not the same as before. So change in delays can basically influence cognitive performance. So we can look at delays in the system, we can simulate dynamics, or we can just look at the spatial length of fiber tracks as a starting point, and potentially better starting point to look at changes in health and disease for brain networks. So it's basically a novel approach to look at brain connectivity. And just one slide on this, last slide. We can also simulate brain development. So in this case, we developed a computer model to simulate the growth of different cortical layers. So we have some cells at the beginning. You have cell division, so the different stages S1 to S7. And then at each stage, you have a probability that the cell dies. Okay, so that's basically early apoptosis, early cell death, or the cell could migrate and then form different cortical layers, and the cell could then die once it has reached the destination. So this sounds like a huge loss, right? I mean, half of the cells are dying. So why are you spending all that energy generating cells and then moving them to a target location and then having them die? It doesn't sound very sensible. But when we look at the simulation, we have the cell division layer here and then the formation of different cortical layers. We find that you need cell death in order to get the same architecture that we see in the human brain and other brains. So if you don't have the cell death parameter in your computer model, you can't regenerate the architecture we see in the brain. So cell death is really essential to get the correct architecture. So with this model, we can reproduce different architectures, a human cortex, rat, mouse, rhesus monkey. So cell death is crucial in enabling a wide range of layer patterns. the model can reproduce brain disorders such as autism, spectrum disorders, polymicrogyria, and subcortical band heterotopia. And there could be a potential use for finding involved genes and predicting drug targets by finding out which processes are going on inside cells to get those patterns we see for brain diseases. Yes? Yeah, so basically you have genetic circuits in the model, and you basically tune the parameters of the genetic circuits. So you change the probability that the cell goes to cell death. And so it's basically have those parameters, and under certain parameters, you can basically reproduce the patterns you see here and the patterns you see in disease as well. Yeah. Okay. So because this is an introduction, there are further books on the topic. Most recent one published in 2020, MIT Press. So that's an overview of brain networks, brain network analysis, looking at spatial features, looking at formation of columns, layers, modules, hubs, and so on, and also looking at changes we see for normal brain development, early development, and late life development, and changes we see for different brain disorders, and changes we see after brain stimulation and for learning. There's the earlier book by Olaf Spons, 2010. This is an introduction to small world networks, so it gives you an introduction to communication across networks and how long distance connections and shortcuts could help communication. This is the first review in the field in 2004, where we explained those features like structural, functional, and effective connectivity. It was funny at that time because Olaf Spons was proposing the term connectome, and we thought it's a bit silly, you know, that you have genomics and proteomics, and so we didn't use the term connectome in that paper, and then Olaf was using it 1 year later in his paper. So thanks for your attention. There was more information on the website there. So yeah, thanks.
Well, I'm not a specialist, but when you make this imaging, right, to detect the fiber, I can imagine that maybe it's easy to detect short fibers, but how accurate are, you know, being not sure that it's long connections actually are true.
Yes. So for the long connections, it's not necessarily the length of the connection that's the main problem. A big problem is curvature. Okay, so if you have a fiber tract that goes like this, you have a very strong curvature of the fiber tract, then the algorithm might just lose it and, you know, just get stuck in the middle, and then you don't have the reconstruction. Thankfully, most of the fiber tracts are more or less a straight line. So very, very few fiber tracts that are highly curved. So curvature is the main problem. It's not necessarily the length of the fiber tract. Yeah. Ramtin? Yeah.
So one question is, about the fact that brain is not optimally wired. So what you're saying here is that it's due to the fact that long connections are used as shortcuts and also to avoid hypersynchronous.
Yes.
But I was wondering, as a speculation interpretation, just honestly makes sense. Is it possible that the brain is not already optimally wired? Because as a, let's say, evolutionary mechanism, like as a defense mechanism, so that if it's needed in case of a damage or something, that it still has some room for optimization. Could that be a possibility as well?
Yeah.
Or
well, that's an interesting point, right? I mean, that you have for neural networks in general, they can absorb failure quite a lot, right? I mean, you can knock out large percentages of the network and still get good function. It could be a way to say that there was more variability in the system on purpose in order to be more flexible.
It's already at the top of optimization. It's damaged, then there's no space.
Yes. I mean, of course, it's always difficult to argue for an optimum. I mean, you always have multiple factors that are influencing the system, and also you wouldn't really say the system is at an optimum. I mean, not everyone has, you know, IQ of 130 or can do long distance running or so. So, you know, so there's variability, and I wouldn't say the system is always an optimal state, but the system is optimized for certain factors, and we can find out which factors are more important than others. Yeah. Hmm. Yeah. Yes. Hmm. Yeah. Yes. So we basically tested for different frequency bands that are normally seen in the brain. So you have like 40 hertz gamma frequency, 20 hertz beta, 10 hertz alpha. And we found consistent results across the frequency bands. It's in a supplementary information, so we don't see a big difference. We're currently testing the link with intelligence, and for intelligence, it seems that gamma and beta are quite indicative of what is going on in the system, but we're still looking at the analysis.
Okay, I think it's 10 o'clock, so we should move on to the next speaker.